A very warm welcome to the talk on structure and functions of smooth endoplasmic reticulum, abbreviated as SER. The endoplasmic reticulum, abbreviated as ER, is a network of membrane enclosed tubules and sacs that extends from the nuclear membrane throughout the cytoplasm. The entire endoplasmic reticulum is enclosed by a continuous membrane and is the largest organelle of most eukaryotic cells. Its membrane may account for about half of all cell membranes, and the space enclosed by the ER, the lumen or cisternal space, may represent about 10% of the total cell volume. There are three contiguous membrane domains within the ER that perform different functions within the cell. The rough endoplasmic reticulum, abbreviated as RER, which is covered by ribosomes on its outer surface, and the transitional endoplasmic reticulum, where vesicles exit to the Golgi apparatus. Both these ER function in protein processing. The smooth endoplasmic reticulum, abbreviated as SER, is not associated with ribosomes and is involved in lipid rather than protein metabolism. The smooth endoplasmic reticulum, that is SER, is devoid of ribosomes. The SER is found in adipose cells, interstitial cells, glycogen storing cells of the liver, gonads, adrenal cortex and leukocytes. The muscle cells are also rich in SER, known as sarcoplasmic reticulum. In the pigmented retinal cells, it exists in the form of tightly packed tubules known as myeloid bodies. The SER forms a continuous system with the rough ER. The smooth ER has a different morphology. In the liver cell, it is made of tubular elements that start at the edge of the cisternae and make a tubular network to the large regions of the cytoplasmic matrix. These fine tubules with some fenestrated smooth membranes are present in glycogen rich regions. In liver cells, the smooth ER has relationship with the Golgi complexes and peroxisomes. These glycogen particles or glycosomes are spheroidal in shape, that is 50 to 200 nanometer, and show an internal structure made of smaller particles of about 20 to 30 nanometer. These are observed under electron microscope and do not correspond directly to glycogen, since this polysaccharide does not stain with the usual electron stains. Glycosomes represent the staining of protein moiety that corresponds to the enzymes of synthesis of glycogen. SER is the region where the synthesis and metabolism of fatty acids and phospholipids occur. Enzymes that modify toxic chemicals or carcinogens to reduce its toxicity are present in SER. These toxic chemicals are secreted out of the body through the formation of vesicles by SER. Now, let's learn the structure of smooth endoplasmic reticulum. The ER is basically a folded structure composed of a network of interconnected disc-like sacs and tubules. All these structures are held in their place by the cytoskeleton. The ER remains bound by a phospholipid membrane. Inside the membrane, a fluid-filled space exists, which is known as cisternal space or lumen. It is found in a variety of cell types and serves different functions in each. It consists of tubules and vesicles that branch, forming a network. In some cells, there are dilated areas like the sacs of rough endoplasmic reticulum. The network of smooth endoplasmic reticulum allows increased surface area for the action or storage of key enzymes and the products of these enzymes. Moving ahead, we discuss the second half of the topic, that is, functions of smooth endoplasmic reticulum. The smooth endoplasmic reticulum has functions in several metabolic processes. It synthesizes lipids, phospholipids and steroids. Cells which secrete these products, such as those in the testes, ovaries and sebaceous glands, have an abundance of smooth endoplasmic reticulum. It also carries out the metabolism of carbohydrates, detoxification of natural metabolism products and of alcohol and drugs, attachment of receptors on cell membrane proteins and steroid metabolism. In muscle cells, it regulates calcium ion concentration. The special functions of smooth endoplasmic reticulum are as described here. Manufacturing. Smooth ER is largely a manufacturing organelle, making and distributing the molecules that a particular type of cell needs. Most smooth ER in both plant and animal cells make lipids or fatty acids, and so cells involved in lipid manufacturing contain many units of this organelle. As an example, animal endocrine cells that use cholesterol to make steroid hormones have a lot of units of SER. In these cells, the SER manufactures hormones. Another function of SER is detoxification. An important function of SER is to detoxify the body from metabolic wastes and drugs. Drug detoxification as well as detoxification of wastes generated in metabolic processes takes place in SER present in the liver cells. It contains enzymes that catalyze a number of reactions that can make lipid soluble drugs and metabolic wastes water soluble so that they can be easily expelled from the body. When a large amount of drugs or alcohol enters the circulatory system, the production of detoxifying enzymes is increased by the liver cells. To perform the function of detoxification, SER doubles its surface area. Once the specific drug or toxin has been eliminated from the body, the excess SER is destroyed by a process called autophagocytosis. SER is also involved in the detoxification of many endogenous and exogenous compounds. The prolonged administration of certain drugs produces an increase, that is hypertrophy, in the SER and the induction of the specific enzymes. In the liver, the SER is specialized for the detoxification of a wide variety of organic molecules, including barbiturates. At least two complex series of enzymes that oxidize these substances are present in SER membranes. If an animal is injected with a large dose of phenobarbital, there is a rapid increase in SER of the liver cells and in their content of these proteins. Carcinogens such as 3-methyl-ecolanthrene and 3,4-benzopyridine are the most potent inducers of drug metabolizing enzymes. The enzymes involved in the detoxification of aromatic hydrocarbons such as 3,4-benzopyrines are aryl hydrolases. Another function of SER is lipid synthesis. In addition to its activities in the processing of secreted and membrane proteins, the ER is the major site at which membrane lipids are synthesized in eukaryotic cells. Because they are extremely hydrophobic, membrane lipids are synthesized in association with already existing cellular membranes rather than in the aqueous environment of the cytosol. Although some lipids are synthesized in association with other membranes, most are synthesized in the ER. They are then transported from the ER to their ultimate destinations, either in vesicles or by carrier proteins. The membranes of eukaryotic cells are composed of three main types of lipids. They are phospholipids, glycolipids and cholesterol. Most of the phospholipids, which are the basic structural components of the membrane, are derived from glycerol. They are synthesized on the cytosolic side of the ER membrane from water-soluble cytosolic precursors. Fatty acids are first transferred from coenzyme A carriers to glycerol 3-phosphate by membrane-bound enzymes, and the resulting phospholipid, that is phosphatidic acid, is inserted into the membrane. Enzymes on the cytosolic phase of the ER membrane then either modify phosphatidic acid or directly catalyze the addition of different polar head groups, resulting in formation of phosphatidyl choline, phosphatidyl serine, phosphatidyl ethanolamine or phosphatidyl inositol. The synthesis of these phospholipids on the cytosolic side of the ER membrane allows the hydrophobic fatty acid chains to remain buried in the membrane while membrane-bound enzymes catalyze their reactions with water-soluble precursors, example CDP choline or citicholine in the cytosol. Because of this topography, however, new phospholipids are added only to the cytosolic half of the ER membrane. To maintain a stable membrane, some of these newly synthesized phospholipids must therefore be transferred to the other, that is, the luminal half of the ER bilayer. This transfer does not occur spontaneously because it requires the passage of a polar head group through the membrane. Instead, membrane proteins called flipases catalyze the rapid translocation of phospholipids across the ER membrane, resulting in even growth of both halves of the bilayer. At least some flipases are phospholipid specific and ATP dependent. In addition to its role in synthesis of the glycerol phospholipids, the ER also serves as the major site of synthesis of two other membrane lipids, that are cholesterol and ceramide. Ceramide is converted to either glycolipids or sphingomyelin, the only membrane phospholipid not derived from glycerol in the Golgi apparatus. The ER is thus responsible for synthesis of either the final products or the precursors of all the major lipids of eukaryotic membranes. Cholesterol and sphingomyelin are important components of lipid rafts. The SER is thus involved in multiple aspects of the metabolism of lipids and lipid-soluble compounds. Synthesis of cholesterol and steroid hormones. SER is rich in cells engaged in lipid metabolism and in those producing steroid hormones. SER is the site of cholesterol synthesis and storage. In the testes, ovary and the adrenal cortex, the SER is involved in the synthesis of steroid hormones. A well-developed SER has been seen in the interstitial cells of the testes of the opossum and guinea pigs, in the adrenal cortex of the rat, and in the corpus luteum of many species. There is a correlation between the amount of SER and the synthetic activity of steroid hormones. Glycogenolysis is a function of SER. SER is related to glycogenolysis. It is engaged in the secretion of glucose to the exterior and is also involved in its protection from the action of glycolytic enzymes in the cytoplasmic matrix. Thus, it regulates the pathway of glucose metabolism in the cell. The withdrawal of glucose from glycogen deposits is mediated by the SER by way of glucose 6 phosphatase. This enzyme catalyzes the dephosphorylation of glucose 6-phosphate to form glucose. The energy stored in glycogen is made available to the body. Large reserves of glycogen are usually found in cells of the liver in the form of small granules attached to the outside of the SER membranes. The function of the glucose 6-phosphatase in the SER membrane is to catalyze the removal of the phosphate group and transfer the glucose formed into the space within the SER. From there, the glucose enters into the bloodstream from where it can be transported to needy cells. Once within the cells, the sugar can be rephosphorylated and metabolized. The enzyme UDPG glycogen transferase is directly involved in the synthesis of glycogen by addition of uridine diphosphate glucose, that is UDPG, to primary glycogen. Ionic gradients. In striated muscle cell, there is an extensive, highly specialized SER called the sarcoplasmic reticulum, which is involved in carrying the stimulus from the nerve cell into the cytoplasm of the muscle fiber. SER is a delicate plexus surrounding the myofibrils. The longitudinal sarcoplasmic tubules form terminal cisternae. A terminal cisterna of each sarcomere and a small transverse tubule constitute a triad which extends over I band. Among the H band, there is a central cisterna which has pores. The central cisterna is formed by the longitudinal sarcoplasmic tubules. In some muscles, the triad lies over the junction of the A and I bends. There are 2 triads per sarcomere. Photoreception. The myeloid bodies in the pigment cells of the retina are modified SER. It is made up of a number of tubules specked over one another. Myeloid bodies are involved in photoreception. Thus, the smooth endoplasmic reticulum plays a crucial role in various metabolic processes. Its existence is important for some life-sustaining processes like protein transportation, carbohydrate and lipid synthesis, and drug detoxification. Hereby we summarize the talk. Endoplasmic reticulum, consisting of tubules, vesicles and sacs, is found in 2 forms, that are rough endoplasmic reticulum, abbreviated as RER, and smooth endoplasmic reticulum, abbreviated as SER. Ribosomes are attached with the RER, which cause certain regions of the cytoplasm to stain with basic dyes. SER lacks ribosomes and hence the name smooth ER. Smooth ER is more tubular than rough ER and forms an interconnecting network subcompartment of ER. It is found evenly distributed throughout the cytoplasm. Under the electron microscope, the SER consists of an interconnected series of convoluted tubules. The SER plays a major role in synthesizing lipids by means of enzymes embedded in these smooth membranes. It produces the phospholipids and cholesterol used in membrane formation, and along with the membrane proteins produced by the rough ER, it can synthesize more membrane for itself for the Golgi complex, the cell membrane, lysosomes and others. In liver cells, the smooth ER contains enzymes for the detoxification of harmful drugs and metabolic byproducts. In the reproductive organs, SER in the cells produces the steroid hormones, testosterone and estrogen. Thank you.